Hi everybody. Today we are going to learn about the fecal streptococcus isolation. Earlier in the previous discussion we had seen that what is fecal streptococcus and what is its role and what is its role as an indicator. How it has been different from the other indicator microbes, that was also been clear to you. But now I'm going to ask you another question. Why do you have to isolate these fecal streptococcus? What is the significance of it? In a study which was being conducted by Dr. Hughes, okay, in UK, he's a microbiologist, and they were trying to conduct a swab analysis of various kitchens, okay, kitchen slabs, and to the astonishment, it was being found that most dangerous organisms and various the most predominantly found organisms which we found there was Pseudomonas aeruginosa as well as fecal streptococcus. Now, where do we have a chance of this to enter? Of course, these organisms do get killed by cooking, but on the slabs of the kitchen, okay, when they took swabs, they found these two organisms. In the case of Pseudomonas, we know it is predominantly found in the soil, and so in the raw vegetables and all that, there is a chance of that to enter into the kitchen. Now, coming to fecal streptococcus, it could be of different reasons. One is it can be by the process of raw meat or anything which before processing, before cooking, it is coming. Or it also could give another wild indication that after going to the uh, washrooms, if we are not washing our hands clearly with soap, what happens? The human, even in human feces, what happens? The fecal streptococcus is there. So the chance of contamination from our own feces onto the kitchen swabs is there. Okay, then another case, another incident of fecal streptococcus contamination was quite interesting. Another study which I noted was they did an analysis of the car in the cars, the children's seats, right? They established children's seats. And among these children's seats, they did certain swabs and they did an analysis. And similarly, it was been found that all the coliforms, most of the coliforms, Enterococcus, Streptococcus fecalis, and in some cases, there were also been found to be certain pathogenic microbes also on the car's seat, in the children's seats. The case was not different in the case of the driver's steering also. Okay, so this fecal streptococcus and all these things can happen to occur in different regions in our day-to-day life. If you go to consider the currencies, the paper currencies that we use, if you do a swab, you can find many of these fecal streptococcus there. Okay, so then if you go to look the sanitary analysis of water, now like you have the water which you take bath in common pools and all that, there are chances of all these fecal streptococci to get contaminated, to contaminate them. And even in the food that we have, fecal streptococcus could be present. So there it becomes essential that the isolation and the identification of this fecal streptococcus becomes relevant. Okay, now let's go into the different methods by which the fecal streptococcus can be isolated. Now, there are different methods by which the fecal streptococci can be isolated. Either you can use the membrane filtration method or the MPN method. I guess you know what is a membrane filtration method. The membrane filtration method is nothing but if you have a sample, let it be a food sample or a water sample. If it's a water sample, you just pass it through a membrane, okay, and all the bacteria within the water sample gets retained onto the membrane. And if it is a food sample, you can homogenize it and then you pass it on through the homogenized sample, and the supernatant of that is taken and it is being passed through the membrane. So it's expected, of course, you will have to centrifuge the food sample after homogenizing so that the supernatant comes. And the supernatant is further passed through the membrane filter. Okay. And then you take this membrane and you put it onto a media which will help in the detection of fecal streptococcus. Then the MPN method, or the most probable number method, will give a estimate, okay, not an accurate but a probable number of fecal streptococcal count in various samples. Even in sewage samples and all that, we use the MPN method. So, in both these methods, let it be the membrane filtration method or in the MPN method, for the presumptive identification of fecal streptococci, we use a agar called the fecal streptococcus agar. So the membrane is either being kept, the membrane after filtration is placed on Streptococcus agar in the case of membrane filtration method, or in the case of MPN method, finally we take the diluted sample and you from the diluted sample you will streak onto the fecal Streptococcus agar plate and incubate it at 37 degrees centigrade for 24 hours. Now, what is the specialty of this media? This media contains a triphenyl tetrasodium chloride. Okay, it's a dye, and what happens is once this is being reduced, it is reduced to form a sand. Okay, and it becomes red in color. So all those fecal streptococci will appear to be, because of the reduction, that is, it's a respiration based process by which this is being using. And so when it gets reduced by the growth of the fecal streptococci, some of this TTC gets converted into formazan, and where all fecal streptococcus is being present, what happens? It becomes red, and a maroon or a pink colony can be seen. So it will enable us to get a presumptive fecal streptococcal count. Coming to the methods to isolate. So after this, you need to have a confirmation of the fecal streptococcus. That is done by subculture on another agar called the bile esculin agar, and you incubate it at 18 hours for 45 degrees centigrade. Now, if fecal streptococci are being present, you will have discrete colonies surrounded by a brown or a black halo, mainly due to the presence of esculin hydrolysis. Okay, this is a bile esculin agar, and here Enterococcus fecalis. If there is a Enterococcus fecalis, what will happen? It is specific for the human gut. Now, this is a So this one, just try to use a marker. The black colored one shows a positive test, as well as the other one, which doesn't have a blackened one. It indicates the control. So if this is get blackened, the media, the initial color would be the brownish color. And if enterophycalis organism, enterophycalis is there, or the fecal streptococcus is there, what happens? It becomes black in color. Now, this is a media. Now, what is the principle regarding this? Here, this is a media which helps us to distinguish between Enterococcus from Streptococcus. I told you Enterococcus fecalis is specific for humans. So, if you want to distinguish between Enterococcus and Streptococcus, you can use this media. You can either use bile esculin media, or sometimes the bile esculin media itself will be supplemented with acid also. Azide is nothing but a substance which will inhibit various other microbes. Okay. So what happens is uh, Enterococcus has an ability to withstand a high percentage of bile. Now, for example, a 40 percentage of bile is being incorporated into this medium, and only Enterococcus can withstand so much amount of bile. But Streptococcus cannot withstand such high percentage of bile, so it will be inhibited in its growth. And what happens is the esculin, the component esculin, is being used up by Enterococcus, and it has been broken down into two components called the esculetin and the glucose. And what happens is S-gluten is further, S-gluten will combine with the what happens. So it will combine with the ion of the media, and it gives a dark brown color or a black complex. So that's how you will be able to identify the enterococcus. So there are mainly two tests which will help us to distinguish the enterococcus from streptococcus. One is the growth on 40% bile and the ability to hydrolyze esculin. And in the case of bile esculin agar, these two properties are being incorporated together into a single plate. And so, after this primary screening, if you want to know whether it is an enterococcus, if you can just inoculate it into the bile esculin agar, and so if you get a blackish colony, it will indicate that it is a enterococcus. So I hope you have understood this, the property of how do you have to isolate it, isolate the fecal streptococcus. The first method that we have is a fecal streptococcus agar, okay, and that has incorporated TPC. And if a fecal streptococcus is being present, what happens? It becomes a reddish or a maroonish color, or a pinkish, not maroonish exactly, but it could be pinkish color. Okay, it depends upon the strain variants would be there. If you get a pinkish colony, you indicate that there is a you can make a presumptive identification of a fecal streptococcus. Further on, by subculturing it onto bile esculinaga, if you get a black colony, it confirms that it is an enterococcus and not a streptococcus. You know, in fecal streptococcus, we have two forms, streptococcus as well as enterococcus. So, hope I think that you understood the fecal streptococcal isolation. Thank you.